Hello everyone, this is Anamika on my channel Easy to Learn. Today we will learn a new topic that is recombinant DNA. First of all, here we learn what is the word recombinant means. We can define it as a relating or a denoting an organism, a cell or a genetic material which are formed by recombination. For more clearance, we can use a similar word that makes more clear about recombinant is reunion. Now we will move on to the next slide and we'll discuss about what is DNA. That is deoxyribonucleic acid. DNA is a molecule that are composed of two polynucleotide chains that coil around each other. As we can see in the diagram that is mentioned in this slide, this structure of DNA was proposed by James Watson and Francis Crick. We can see here that this DNA it contains sugar phosphate. There is also the presence of nitrogenous bases that are guanine, cytosine, thymine, and adenine. Now we will go through the definition for recombinant that makes you more easier uh, to be aware of the topic. Recombinant DNA is the joining together of DNA molecules from different organisms and inserting it into a host organism to produce a new genetic combinations that are of value in the field of science, medicine, agriculture, and industrial fields. We will go through some of its objectives. That it helps in identification of genes, it can isolate the genes, and also helps in the modification of genes. Here the isolating the genes, what does it mean? We can say it as the lack of interbreeding or genetic mixing between organisms of the same species. This may also result in speciation. This technology also manufactures large quantities of specific gene products such as hormones, vaccines, and other biological agents. I have mentioned here the technique for the recombinant DNA technology. Let's have a look. We can define it as that when a scientist isolate a gene of interest from one organism and insert it into another vector, thereby forming a recombinant DNA molecule. Here the large quantities of the gene of interest, that is also called as the product of the gene, are produced when the bacteria, that is the vector carrier, multiplies to increase in numbers. So one of the suitable example is reproducing insulin for patients with diabetes mellitus, that is type 2 diabetes, as this diabetes is so much common in our country. Here we will go through some of the applications of recombinant DNA technology, the treatment strategies that also improved by developing diagnostic kits, monitoring devices. Next is hepatitis B infection is also controlled with the use of a recombinant hepatitis B vaccine. Recombinant DNA technology is also beneficial for us not only in the field of medicine but also it plays a vital role in food and nutrition. I hope this video will help you and make you easy to learn. Please like, share and subscribe my channel. Thank you.